we are at visual system, right? We are at visual system, and today is the last class, that is part 3 of visual system. So a little recap. So we had started with the normal anatomy of the eye. Then we have discussed the location of fovea, macula, and the location of the optic disc and normal physiological cupping and the blind spot. Next, we have discussed the layers of retina, 10 layers of retina, very important question which is frequently asked in viva examination. But the major cell layer is your 3 cell layer, that is rod cell and cone cell layer. Next is the bipolar cell layer, which is the bipolar neuron, and next is the ganglion cell layer, right? Okay, now next we had discussed details about rod cell and cone cell, right? We had discussed details about detailed structure and function of rod cell and cone cell. There are 120 millions of rod cell and 80 to 90 millions of cone cells. And each cell is divided into few parts, that is outer segment connecting cilium, inner segment nuclear region, and synaptic terminal, right? So here is the picture of rod cell and cone cell which are rich in mitochondria, right? So next, detailed structure of rod cell and cone cell, we are not moving into it. Now the function of rod, as you all know, that rod cells are responsible for vision in dark, that is scotopic vision, and the cone cell, they are basically responsible for visual acuity. And the cone cells, which are responsible mainly for color vision, and there are 3 different types of cone cell, you know, which is sensitive to blue light, green light, and red light. So that is short wavelength cones, medium wavelength cones, and long wavelength cones. These 3 types of cones, presence of these 3 types of cones, and their responsibility in detecting different types of colors is also called the Young-Helmholtz theory, right? Okay, now the function of cones, it is basically for color vision or photopic vision, okay, but visual acuity will be less. Now here is the difference between rod cell and cone cells. So remember the quantity, 90 to 120 million of rod cells and cone cells 4.5 to 6 millions. Now the functions of Muller cells, horizontal cells, and amacrine cells, these are all supporting cells, and these are helping in the metabolism of the layers. These are supporting cells, scaffolding cells, right? And also helps in metabolism and supplying nutrition to the retina. So Muller cell, horizontal cells, and amacrine cells. Now next we had discussed the visual pathway. You all know that it starts from the retina. So horizontally you have the temporal retina and the nasal retina, and both the retina is having one optic nerve, okay. So optic nerve, it decussates in optic chiasma, where the nasal fibers from the opposite sides, they cross each other, whereas the temporal fibers remain on the same side. Now after optic chiasma, it is optic tract. Optic tract goes to lateral geniculate body or lateral geniculate nucleus, from lateral geniculate nucleus to the optic radiation, and optic radiation moves to primary optic cortex or primary visual cortex, which is also called area 17, or it is also called the area V1, okay, area 17, broadband area 17, or also called V1, okay. In the optic radiation, you have 2 different types of loops. One is Meyer's loop or the temporal loop, and another is parietal loop or the dorsal loop, right? Okay. Now the visual pathway. Now another thing is, in the lateral geniculate nucleus, you have 6 layers. Okay, 1, 2, 3, 4, 5, 6 layers in the lateral geniculate nucleus. Layer 1 and 2 are responsible for sensing vision. Layer 1 and 2 are responsible for sensing color vision, motion, that is M for motion, okay, magnocellular pathway and parvocellular pathway. Layer 1 and 2 forms the magnocellular pathway, and layer 3 to 6 forms the parvocellular pathway. Okay, so magnocellular pathway and parvocellular pathway. Magnocellular pathway is for motion, right? So sensing motion, okay. Now next, the visual field effect, which is the most important clinical things from this from the slides, okay. So lesion in the macula, it will cause central vision loss. Lesion in the optic nerve, it will cause left anopia. Lesion in the optic chiasma, it will cause bitemporal hemianopia. Lesion in the optic tract, left optic tract, it will cause right homonymous hemianopia. Meyer's loop lesion. So in case of lesion in the Mayer's loop, you will have, the patient will have pie in the sky type of scotoma. Okay, pie in the sky type of scotoma. So similarly, where is your nasal fibers and temporal fibers in the visual field, they are crossing each other. That means with your nasal retina, you see temporal field of vision, and with your temporal retina, you see the nasal field of vision. Similarly, if we draw a horizontal line, from within the retina, right? So your upper retina sees the lower visual field, and the lower retina sees the upper visual field, right? Okay, so crossing is there, temporal and nasal, and also upper retina and lower retina. Okay, এই জিনিসটা ক্লিয়ার হলো সবার? So here is your nasal retina, here is your temporal retina, nasal retina and temporal retina. This is left eye and this is right eye, right? Okay, so temporal retina will see the nasal area, and nasal retina will see the temporal field of vision. Okay, so this is one thing. Similarly, if we see like this is upper half of the retina and this is lower half of the retina. So upper retina sees the lower visual field and lower retina sees the upper visual field. Clear? Here is the visual field. Okay, so this crossing is there. Okay, so once the lesion is in the temporal loop or Mayer's loop, it is pie in the sky, and once the lesion in the parietal loop, it is pie in the ground. Or when it is lesion in the visual cortex, it is again homonymous hemianopia, but there is central sparing because of one-to-one representation of macula. Okay, now next we have talked about phototransduction mechanism, right? Next we had talked about phototransduction mechanism. That means how light energy is converted into an electrical energy. Light energy is converted into electrical energy and gives a sensation of vision, right? We had discussed in details, we are not moving into it. Remember, here we have introduced two proteins, one is rhodopsin and another is transducin. Rhodopsin or conopsin or photopsin, it is the name of the G protein coupled receptor. Okay, G protein coupled receptor or GPCR, which rotates 7 times within the cell membrane, right? And transducin is the another name for G protein. Okay, so here this 7 transmembrane protein is rhodopsin, and this is 11 cis retinal, which is converted into all trans retinal during phototransduction mechanism. We are not moving into details because everything is discussed. Okay. Okay, we have also talked about the presence of 2 different types of bipolar cell and 2 different types of ganglion cell, which is responsible for taking hyperpolarization into action potential. So unlike all other cell, it is the retinal pigment epithelium cells or rod cell or cone cells where the primary event is hyperpolarization and not depolarization. Okay, this hyperpolarization is converted into depolarization and action potential into the ganglion cell because of the presence of 2 different types of bipolar cell and 2 different types of ganglion cell. Okay, on bipolar cell and off bipolar cell, similarly on ganglion cell and off ganglion cells. Okay, 2 different types of ganglion cell, on center off surround and off center on surround. Okay, next we have talked about dark adaptation, and there is a curve of dark adaptation. So cones act first, and this is rod cone break. So initially when you enter into a dark room, sensitivity first increases for the cones, but after 5 minutes it goes into play too, then rods come into action, right? So then rods take action, the remaining part is due to rods, right? And finally your dark adaptation is basically due to rod cells and not the cone cell, although it is started by cone cells. Okay. Now, when the patient's dark adaptation will be defective, it is due to vitamin A deficiency, retinitis pigmentosa, aging, and glaucoma and other retinal diseases. Now, properties of visual cortex, we have talked about 4 different types of property. One is retinotropic mapping, that is point to point mapping. Every point of retina is represented in the visual cortex, just like your sensory homunculus and motor homunculus in the visual cortex or area 17 or area V1, each point of the retina is pointed. Next is orientation selectivity, where we have talked about 2 different types of cell. One is simple cell, another is complex cell. Simple cells are sensitive to lines and complex cells are sensitive to motions. Okay, then ocular dominance column, alternating stripes receiving input from the left eye and right eye. Feature extraction. So during talking about feature extraction, we have talked about blobs and interblobs. Blobs are responsible for color processing. They are specialized cells in the visual cortex which are rich in cytochrome oxidase, and interblob region are responsible for orientation and form processing. Next is plasticity. Cortical plasticity, we had discussed neuronal plasticity in previous classes. What is synaptic plasticity? Due to history of previous discharge, right? The strength of a synapse can be changed, right? The strength of a synapse can be changed due to history of previous discharge. Okay, so just like while discussing memory, we have talked about long-term potentiation and long-term depression, where the strength of synapse was increasing in long-term potentiation and the strength of synapse was decreasing in long-term depression, right? So similarly here in visual cortex, it is also related to memory. The neuronal synapse strength can be changed due to history of previous discharge, right? So visual cortex, your visual cortex similarly have the plasticity, neuronal plasticity. That means the change in the strength of the synapse. Okay, there are other visual cortex, not very essential for you. Secondary visual cortex V2, then V3, V4 and V5. Okay, higher order visual streams. Color vision, again important short note for you, color vision. So again you will tell there are 3 different types of cones. S cone, M cone and L cone for sensing blue light, green light and red light, and the presence of 3 different types of cones is also called the Young-Helmholtz theory, right? Young-Helmholtz theory. Okay, now the last slide we talked yesterday, is not yesterday, in our previous class is congenital deficiencies of 1 or more cones. If it is deficiency of L cones, it is protanopia. If it is deficiency of M cone, it is called deuteranopia, and it is deficiency of S cone, it is called tritanopia, right? Okay, pupillary light reflex pathway. Again, this slide is important for today's discussion. Okay, so we had talked about visual pathway where optic tract, okay, optic tract goes to lateral geniculate body, but there are some nasal fibers, right? There are some nasal fibers which just bypass the lateral geniculate body and goes to the midbrain and ends in pretractal nucleus in the midbrain, right? Okay, so these are the fibers, these are the nasal fibers, this blue color and this red color, they are crossing each other, and see, they are just bypassing lateral geniculate body, not ending in the lateral geniculate body, going straight to pretractal nucleus in the midbrain, and from pretractal nucleus they are moving to bilateral Edinger-Westphal nuclei. So this is the basis of direct light reflex and consensual light reflex. When light is shown into one eye, both pupil will constrict, because pretractal nucleus has convergence into or divergence into both the Edinger-Westphal nucleus, both right and left Edinger-Westphal nucleus. So this is actually the basis of consensual light reflex, right? Okay, now further, what is the efferent pathway of pupillary light reflex? From pretractal nucleus to bilateral Edinger-Westphal nucleus, from bilateral Edinger-Westphal nucleus to ciliary ganglion through oculomotor nerve or third cranial nerve, from ciliary ganglion through short ciliary nerve to sphincter pupillae. Okay, through sphincter to the sphincter pupillae, which causes meiosis or constriction of pupil. Okay, So light in either retina sends a signal via cranial nerve 2 to pretectal nuclei in the midbrain that activates bilateral Edinger-Westphal nuclei. Pupils constrict bilaterally, which is the basis of direct and consensual right reflex, and the result is illumination of one eye result in bilateral pupillary constriction. Okay, now starting where we had ended in our last class. Answer. So it is about ocular movement. You know, superior oblique, inferior oblique, medial rectus, lateral rectus, superior rectus and inferior rectus. So these are 6 muscles for ocular movement. So here the question is, a boy has been scolded by his mother, and his gaze is down and towards right. Down and towards right. Okay, down and towards right. Answer. Inferior rectus, superior oblique. How many of you are telling inferior rectus and superior oblique? Inferior rectus and superior oblique. Okay. Okay. Any other option you think? D. D means lateral rectus, superior oblique. Okay. Okay. So let's discuss this. Okay. So here is the complete function of the extraocular muscles. Okay, complete function. If you remember this picture, you will be able to tell all the functions of extraocular muscle. Okay. So what is the function of lateral rectus? Abduction. Okay. The function of lateral rectus is abduction. Okay. What is the function of medial rectus? Adduction. Okay. So this is nasal side, this is temporal side, this is right eye, this is left eye. Okay. So C it is right, R for right, L for left, right? Okay. This is nasal side and this is temporal side. Okay. So medial rectus is for adduction and lateral rectus is for abduction. This function is very clear to everyone. Okay. Now coming to superior rectus and inferior rectus. Okay. So superior rectus, elevation. Okay. Elevation, and also this is superior rectus. Okay. That means elevation, not only elevation, this is, yes, extorsion, or you can also say elevation with abduction. Okay. Elevation with abduction. So superior rectus, again see here, elevation and elevation with abduction. Okay. Elevation and elevation with abduction. Now superior oblique. Superior oblique is adduction, or you can say depression in adduction. Okay, depression in adduction. Superior oblique, depression in adduction. Clear? Depression in adduction. Remember, because if you see anatomy, if you dissect eyeball, you will see the muscle just rotates. So that is why even if it is superior oblique, the name is superior, but it is depression while in adduction. Okay, depression while in adduction. And here it is inferior oblique, it is elevation in adduction. Okay, elevation in adduction. So actions are opposite, right? Superior rectus and inferior oblique, their actions are opposite. Superior rectus and inferior oblique. Okay, inferior oblique causes elevation in adduction, and superior rectus causes elevation in abduction. Clear? Okay. So if you are able to remember this picture, everything will be clear to you. Okay. Now again come back to the question. Tell your answer now. So it is clearly for right eye it is inferior rectus, for right eye it is inferior rectus, and for left eye it is superior oblique. Okay, so that was option B. Okay, so option B is the correct answer. Right eye inferior rectus, left eye superior oblique. One is answer B, right eye inferior rectus, left eye superior oblique. Okay, so most of you have done it correctly. This was a direct question from USMLE, just copied. So here we summarize the function of all the extraocular muscle. Okay, medial rectus adduction, lateral rectus abduction, nothing else. Rectus elevation and adduction, intorsion, inferior rectus depression, adduction, extorsion, superior oblique, intorsion, then abduction and depression in adduction, and inferior oblique elevation in adduction. Okay, superior oblique depression in adduction, inferior oblique elevation in adduction. Okay, these two functions are important. Okay, now pupillary control. When pupil constricts, we call it miosis, and when pupil dilates, we call it mydriasis. Okay, when pupil constrict, we call it miosis, and when pupil dilates, we call it mydriasis. So, what is the mechanism? Miosis is a parasympathetic event, and mydriasis is a sympathetic event. That means when parasympathetic system will be active, acetylcholine mediated, there will be miosis. Okay, miosis is parasympathetic, and what is the pathway? First neuron is definitely, everyone knows, it is coming from the Edinger-Westphal nucleus, then to the ciliary ganglion via cranial nerve tree, and the second neuron is the short ciliary nerves, which innervates the sphincter pupillae muscle. So short nerves shorten the pupil diameter. Here is the mnemonic. Okay, short nerve shorten the pupil diameter. Okay, first nerve is the third cranial nerve, which is coming from the Edinger-Westphal nuclei to the ciliary ganglion, and the second nerve is the short ciliary nerve. Okay, it is the pathway of miosis. This is how pupillary constriction takes place. You also know this from the pathway of pupillary light reflex. Then mydriasis. Mydriasis is a sympathetic phenomenon which is mediated by sympathetic neurotransmitter, that is adrenaline. So pupillary dilation mediated by sympathetic, and the first neuron, it comes from hypothalamus to the ciliospinal center, which is located in the C8 to T1 sympathetic ganglia, right? And the second neuron, it exits from T1, which is the superior cervical ganglion, and the third neuron comes from the plexus around the internal carotid, right? So here are, it is the 3 neuron mediated pathway, and meiosis is the 2 neuron mediated pathway, okay? Okay, now RAPD or relative afferent pupillary defect. It is again a clinical application of pupillary light reflex. Okay. So first see what will happen if there is a defect in the afferent pathway. What will happen in the defect in the afferent pathway? It frequently happens in optic neuritis, when the optic nerve is inflamed. Okay. When the optic nerve is inflamed. Okay. So RAPD is defect in the afferent pathway or sensory input of the parietal of the pupillary light reflex. Okay. So if you remember the pathway of pupillary light reflex, the initial path is same as that of visual pathway. Okay. It starts from the retina, then optic nerve, then optic chiasma, then optic tract. Okay. So this pathway is similar in pupillary light reflex and in the visual pathway. Now what will happen if the afferent pathway is defective? That means either retina is defective or optic nerve is defective, right? Okay, so RAPD is the defect in the afferent pathway or the sensory input of the pupillary light reflex. It means less light information is getting to the brain from the affected eye. Okay? তাহলে affected eye তে যদি একদিকের optic nerve খারাপ থাকে, right? একদিকের optic nerve খারাপ থাকে. Okay, say for example my right eye optic nerve is damaged, that is optic neuritis or something, some disease process is there which is causing inflammation in the optic disc or optic nerve. Okay, so if you show light to my right eye, what will happen? There will not be any direct reflex, there will not be any consensual light reflex, right? Clear? So once right optic nerve is injured, that means optic neuritis. So if you show light to my right eye, there will be no direct reflex, there will be no consensual reflex, because consensual reflex pathway is also coming from right optic nerve. Clear? Okay. So it is called relative because we compare both the eyes, and the efferent pathway are intact, so pupil can still contract. Okay, pupil can still constrict. Okay, it happens in optic neuritis. The major diagnosis is optic neuritis. তার মানে কি হবে, তুমি পেশেন্টের একটা চোখ খারাপ আর একটা চোখ ভালো, তো প্রথমে আমি ভালো চোখটাতে যেটা হেলদি আই আছে তাতে যদি লাইট ফেলি তাহলে কি হবে, হ্যাঁ ডিরেক্ট কনসেনসুয়াল দুটোই হবে যদি ভালো চোখে ফেলি। ওকে, ডিরেক্ট কনসেনসুয়াল দুটোই হবে। তারপরে যদি আমি ড্যামেজ আইতে অর্থাৎ ডিজিজড আইতে লাইট ফেলি তখন কি হবে, ইনস্টিড অফ কনস্ট্রিকশন ওটা একই রকম থাকবে বা ডাইলেট হয়ে যাবে, রাইট? ওকে, সো ইট ইজ ডিউ টু আরএপিডি কখন হয়, পার্শিয়াল ড্যামেজ টু দি অ্যান্টেরিয়ার টু দি অপটিক chiasma, অপটিক chiasmaর আগে যদি ড্যামেজ হয় মানে অপটিক নার্ভ বা রেটিনাতে যদি ড্যামেজ হয়। ওকে, লেস ইনপুট ফ্রম দি অ্যাফেক্টেড আই। So how to test RAPD, relative apparent pupillary defect? How to test it? Okay, we do it by swinging flashlight test. It is called swinging flashlight test. Okay, and the most important clinical test to detect RAPD. So in a dim room, ask the patient to fixate at a distant object. At a distant object, a patient fix korbe. Now shine the light to the normal eye first. Okay, shine the eye to the normal eye first. প্রথমে normal eye তে আমি light ফেলবো, right? Okay, both the pupil will constrict briskly, you know, the direct and consensual light reflex. Now suddenly I will swing light to the affected eye or the diseased eye. Okay, normal eye থেকে suddenly আমি swing করে লাইটটা diseased eye তে নিয়ে যাব, right? So what will happen, because of the afferent input is reduced, both pupils will paradoxically dilate, okay, or constrict less. This is the hallmark of RAPD, and this is called swinging flashlight test. Okay, যদি নরমাল হতো তাহলে দুদিকে লাইট ফেললে ইকুয়ালি কনস্ট্রিকশন হতো, বাট ইন কেস অফ RAPD there is afferent input is reduced in the diseased eye, so the pupils will paradoxically dilate, and it happens in optic neuritis. The major diagnosis is optic neuritis. Common causes, okay, unilateral or asymmetric optic nerve or retinal disease, optic nerve lesion. Major diagnosis আমি আগে বলে দিয়েছি, optic neuritis. It frequently happens in multiple sclerosis. You don't know what is multiple sclerosis, gradually you will know. Okay, ischemic optic neuropathy and optic nerve compression due to a tumor or space occupying lesion. Okay, now the final part. With this I will end today's class, and also I will end vision. Okay, so it is internuclear ophthalmoplegia. এটা আগেও একবার পড়ানো হয়েছে, internuclear ophthalmoplegia during discussion on vestibular system. Okay, it is a conceptual topic, internuclear ophthalmoplegia, right? So to know internuclear ophthalmoplegia, what the disease is, internuclear ophthalmoplegia or INO, you need to know what is MLF, medial longitudinal fasciculus. Okay, you need to know what is medial longitudinal fasciculus, right? So see what is medial longitudinal fasciculus. So here is the nuclei for sixth cranial nerve. Here is the nuclei. This yellow color nuclei is sixth cranial nerve. Okay, sixth cranial nerve, it supplies which extraocular muscle? Sixth cranial nerve, lateral rectus. Okay, okay. So if you are asked to look your right, if you are asked to look your right, okay, your right, so your right eye lateral rectus will work and left eye medial rectus will work, right? Okay, so it is called conjugate eye movement. This phenomenon is called conjugate eye movement. একটা আইয়ের lateral rectus কাজ করবে আর একটা আইয়ের medial rectus কাজ করবে, right? This is called conjugate eye movement. But you know that lateral rectus is supplied by sixth cranial nerve and medial rectus is supplied by third cranial nerve. So that essentially means inside your brain or brainstem, there must be a circuitry that connects third cranial nerve and sixth cranial nerve. Okay, there must be a connection between third cranial nerve and sixth cranial nerve, and that should happen very fast to help you in conjugate movement, so that suddenly you can look at your right, and suddenly you can look at your left. Okay, so once you look at your right lateral rectus and left medial rectus working. Similarly, if you are looking at left, your left lateral rectus and right medial rectus are working, right? Okay, so that means একটা সাইডের সিক্সথ ক্রেনিয়াল নার্ভ নিউক্লিয়াসের সঙ্গে অন্য সাইডের থার্ড ক্রেনিয়াল নার্ভ নিউক্লিয়াসের there must be a connection. Okay, and that connection is your medial longitudinal fasciculus, this green color. Okay, this is called medial longitudinal fasciculus. Okay, so what is happening, you see, so this is sixth cranial nerve. Okay, sixth cranial nerve is going to sixth cranial nuclei, and from the nuclei sixth cranial nerve goes to lateral rectus, cranial nerve 6, and this sixth cranial nerve is connected through right medial longitudinal fasciculus to the opposite oculomotor nucleus or third cranial nerve nucleus, and this third cranial nerve is again connected to the medial rectus. Okay, okay, tale right sixth cranial nerve is connected to right sixth cranial nerve nuclei is connected to left third cranial nerve nuclei through right medial longitudinal fasciculus, and left abducent nuclei or left sixth nerve nuclei is connected to এখানে খালি রাইটটাই দেখানো আছে ছবিতে, রাইট? সো this sixth cranial nerve nuclei, left sixth cranial nerve nuclei will be connected to right oculomotor nuclei through left MLF or left medial longitudinal fasciculus. Clear? Okay, and the problem happens when this medial longitudinal fasciculus is damaged. Okay, due to stroke or any hemorrhage or any tumor or any space occupying lesion, when this medial longitudinal fiber is damaged, what will happen? So once we ask the patient having damaged medial longitudinal fasciculus, okay, with a damaged medial longitudinal fasciculus, so I am discussing asymmetric, or that means unilateral medial longitudinal fasciculus, because both side will be having a complex event, right? So first understand unilateral or one-sided medial longitudinal fasciculus damage. What will happen? Okay, so if I ask the patient to look at his right, okay, I am asking the patient to look at his right. Okay, so your right is this side. Okay, so I am asking the patient to look at right. So definitely sixth cranial nerve nuclei is okay for the patient. So patient's right eye will move towards right. That means lateral rectus will work perfectly. Okay, but the medial rectus will not, because the connection from sixth cranial nerve nuclei to the opposite third cranial nerve nuclei is lost. Okay, so right eye abduction. Okay, right eye abduction hobe, আর left eye adduction হওয়ার কথা কিন্তু হবে না। ওকে, left eye adduction হওয়ার কথা কিন্তু হবে না। ওকে, so internuclear ophthalmoplegia is a phenomenon where there is adduction failure, okay, adduction failure of the opposite eye, and the unilateral eye or the ipsilateral eye, there will be abduction with nystagmus. Okay, abduction with nystagmus. Nystagmus কেন হবে? Because this cranial nerve 6 is firing vigorously to activate cranial nerve 3, opposite cranial nerve 3. But it is not happening because medial longitudinal fasciculus is already damaged, right? Okay, so this is why there will be, due to repeated firing of the sixth cranial nerve towards medial longitudinal fasciculus, there will be nystagmus in abduction. Okay, there will be nystagmus in abduction, but in the opposite eye there will be adduction failure. এবার আমরা একটু পড়ে নিই কি লেখা আছে। Okay, so internuclear ophthalmoplegia, we need to know the medial longitudinal fasciculus. Medial longitudinal fasciculus is very vulnerable to damage because this medial longitudinal fasciculus is a high myelinated fibers. Okay, these are collection of highly myelinated fibers. Why highly myelinated? Because speed of action potential traveling through medial longitudinal fasciculus has to be increased because your conjugate eye movement is so fast, right? So it is to help you in your conjugate eye movement, right? So medial longitudinal fasciculus, it is a pair of tracts that interconnect cranial nerve 6 and opposite cranial nerve 3 nuclei. Okay, it coordinates both the eyes to move in the same horizontal direction. It is highly myelinated, must communicate it quickly, so your eyes move at the same time. Okay, মানে lateral rectus আর medial rectus যাতে একই টাইমে মুভ করে তার জন্য medial longitudinal fasciculus is highly myelinated and the speed is very high. So lesion may be unilateral or bilateral. Lesion in the medial longitudinal fasciculus, medial longitudinal fasciculus এ যদি ইনজুরি হয় সেটাকেই আমরা বলি internuclear ophthalmoplegia, okay, or INO, internuclear ophthalmoplegia. It is a conjugate horizontal eye movement palsy. Okay, conjugate horizontal gaze palsy or conjugate horizontal eye movement palsy. Okay, lack of communication such that when cranial nerve 6 nucleus activates ipsilateral lateral rectus, contralateral cranial nerve 3 nucleus does not stimulate the medial rectus to contract. Okay, medial rectus contract করতে পারছে না. Okay, তাহলে ultimately effect কি হবে? Abducting eye, যেটা cranial nerve 6 stimulate করছে, abducting eye will show nystagmus because cranial nerve 6 overfires to stimulate cranial nerve 3. Okay, cranial nerve 6 is overfiring to stimulate cranial nerve 3. Convergence will remain normal. So normally when looking left nucleus of cranial nerve 6 fires, which contracts the left lateral rectus and stimulates contralateral or right nucleus of the cranial nerve 3 via medial longitudinal fibers to contract the right medial rectus. Okay, so remember this line. Once there is INO, internuclear ophthalmoplegia, ipsilateral adduction failure and nystagmus in the opposite. Ipsilateral adduction failure, okay, ipsilateral adduction failure and contralateral abduction with nystagmus. Okay,